I am excited to have Dr. Eric Green, the director of National Human Genomics Research Institute of NIH. So those students who is taking camp this week and now yesterday you heard about NIH, and that NIH is one of the big branch of the NIH. He is the director. So in other words, he guide, direct human genome research where to go. United States did research also direct worldwide internationally. So basically, he's going to see the future, what is going to, and also he knows from the start and end of the first human genome project. So I am honored and so happy to have Dr. Green in this talk. Dr. Green, would you start talk?
Absolutely. So first, even before I get to my slides, let me start off by wishing you all the best. I hope everybody is safe and staying healthy during this very challenging time related to the pandemic. I think a program like this is a great thing to be doing, especially because you're doing it virtually. What a great way to spend time when you can't really get out and about too much. And the fact that you're all here wanting to learn science is, I think, very uplifting. I also want to say to Dr. Lee, congratulations to you for putting this together and for organizing it. I'm delighted to participate and to do this. In fact, to be honest with you, this is probably going to be the highlight of my week, which probably says something about all the things I have to deal with this week, which aren't nearly as much fun. So I am thrilled to be here and really looking forward to talking to you about this exciting field of genomics and also to answer your questions. So let me try, I'm going to share my screen now. And if I do it right, you should now all see my first slide. Could somebody nod and confirm with me that you can see my first slide? Yeah, okay, great. So I have been involved in this field of genomics since the field actually began, essentially. And what I want to do today is really talk about this journey that I have been involved with, and that involves some historical background that'll be important, tell you a little bit about what's going on now, and also take you a little bit into the future. And all of this is a journey aiming to advance human health. It was actually interesting, and I knew that, or I was told that you were given this editorial. I think this is where Dr. Lee came to learn about me directly by reading this. And this is an editorial that I wrote in this journal, or this magazine called The American Biology Teacher, and I wrote exactly 2 years ago. And it was written as sort of a, because they asked me to do it as a letter to my daughter, who was then graduating high school, who's now finishing her, or just has finished her second year at University of Virginia. And it was just sort of a past, present, and future explanation, because sort of I had seen the arc of genomics sort of evolve considerably during her lifetime. And in rereading this last night, because admittedly I hadn't read this for a couple of years, and rereading it actually is going to completely parallel what I'm going to discuss in my talk to you. I'm just going to add a lot more detail than what was available in that one-page piece from 2 years ago. And more than anything, I'm not going to go into a whole lot of detail about any one thing. I want to really tell you a story, because I think the story of genomics is so captivating, so important, and I hope will be very enticing to you, because if you're interested in science, if you're interested in medicine, this is a field that is not only growing, but is incredibly relevant to almost all areas of biology and all areas of medicine, increasingly. Just by way of background, and I realize I'm dealing with a very heterogeneous audience. Some of you have probably taken advanced biology classes. Some of you have just taken introductory biology classes. So I realize that everybody's coming at this maybe with a slightly different background. So let's just get everybody grounded in some pretty simple basics. You know, the human body, like all living systems, consists of cells. The human body has several tens of trillions of cells. What's very interesting about biological systems like the human body is that every one of those cells operates off the exact same blueprint, the fundamental blueprint that is responsible for determining how the organism gets built and how it operates. It's both a blueprint, it's actually also an operating manual. But every cell has the identical blueprint, and yet many of our cells do very different things depending upon if it's a brain cell or if it's a muscle cell and so forth. So for decades, actually centuries, we were intrigued by understanding what could that blueprint be. What could be that blueprint that really provides all the information necessary for developing and operating a human? Well, we learned when the microscope was invented that in the middle of cells were these structures called nuclei, and in the nucleus of cells are housed these structures called chromosomes. And it turns out that chromosomes are actually the suitcases that carry this genetic blueprint, or if you will, this blueprint from cell to cell, from generation to generation. And ultimately, we figured out that the information molecule of this blueprint, fundamental to all living systems, was this molecule of DNA. And DNA was, once it was recognized last century that it was the information molecule of living systems, a tremendous amount of attention went to try to figure out everything we could learn about DNA, and of course, in particular, about human DNA. The number one discovery, in my opinion, in biology last century was this discovery of Jim Watson and Francis Crick. The famous double helical structure of DNA was determined by them in 1953. That big iconic double helix, it was so critical to figure out the structure of DNA, because until they figured out the structure, we didn't really understand how was it that DNA could be the information molecule that carries all the blueprint information from one cell to the next, from one generation to the next. But that double helical structure was the final piece of information that gave us the clue about how it is that it beautifully choreographs this information flow from cell to cell, from generation to generation. That was the 1950s. By the 1960s, the famous, and if anybody taken a biology class, you've seen this table. It's the famous lookup table of how is it that DNA in its letters end up coding the information for making proteins. Proteins are the building blocks of cells and tissues. And it's that famous codons, those three letters that each code for a different amino acid. That was discovered actually right here in Bethesda, Maryland, where I work at the National Institutes of Health. That's where it was discovered in the 1960s. By the time I got to high school in particular, and then in particular when I was in college, it was all part of what was called the molecular biology revolution. When we figured out how to clone DNA, we figured out how to manipulate DNA, and when we got better and better at actually reading the letters of DNA. So the discovery of DNA double helix, and then discovery of how we could decode it to make proteins, and then this surge of technical abilities to be able to study DNA in the laboratory, clone it, sequence it, manipulate it, all sort of led to this idea that, wow, maybe we are positioned to actually read out all of human DNA once and for all. And of course, remember, the DNA alphabet is not terribly complicated. It's actually simpler than proteins. It turns out there's only 4 building blocks, as you know, in DNA. We even take a shortcut and we don't even talk about the 4 chemicals. We talk about the letters. We talk about A, G, T, and C. And it turns out that human DNA, all the DNA in an organism is called a genome. But the whole human genome isn't infinite. It's just big. And it's big in that it consists of 3 billion bases, or 3 billion of these A, G, T's, and C's. In other words, it's 3 billion letters. And while 3 billion is a big number, it wasn't impossible to imagine that someday we could actually read out 3 billion letters if we got really good at reading DNA. That, my friends, in the late 1980s, led to this idea of, let's once and for all read out the human blueprint. Let's figure out the order of these 3 billion G's, A's, T's, and C's, and then we will have in hand a sequence of the human genome, which will give us fundamental insights about the blueprint necessary for making and operating a human being. This was organized into something called the field of genomics, because the field of genomics didn't even exist until 1987. In fact, you will never find the word genomics until 1987, until a new journal got called Genomics, where they sort of defined the field. By the way, I'll just pause for a minute and point out, I grew up in the Midwest. I was born and raised in St. Louis. I went to college at University of Wisconsin, and then I came back to St. Louis to Washington University, where I was in their MD PhD program. I got both a medical degree and a PhD doctoral degree. And I graduated in 1987. That was when I graduated with my MD PhD. So just think about it. I am now in charge of the organization in the United States that leads genomics research in the US. And yet I never heard the word genomics once in medical school or graduate school. because the word hadn't been invented yet. So there I was with an MD and a PhD learning the word genomics for the first time. And basically that was how I made my career, was in genomics. It's an interesting sidelight in that it illustrates that you really are a lifelong learner. Even if you're given an MD and a PhD, you're still learning. And I never learned any genomics until I got out of medical school and graduate school, because the word didn't even exist. Okay, back to the story of genomics. So the field was created in 1987, a very young field compared to fields like genetics or fields like cell biology or physics. But genomics was born in 1987, and the reason a field in part was named was to galvanize the interest and organize efforts to tackle what I was just alluding to, the ability to actually develop a program that would allow us to read out the complete human genome sequence, all 3 billion letters. This was organized as an international effort called the Human Genome Project, which all of you are so young, you read about in your history books. But there I was, just graduated from medical school. I was in the middle of a residency, and I got involved in this project on the very first day, actually, in October 1 of 1990. And the project was very unusual. It was big, it was international, there was multiple countries involved, thousands of investigators around the world, because it was recognized not just the United States human genome, this was humanity's human genome. We needed to do this internationally and share all the information and really enable our understanding of human biology for all applications around the world. This project was amazingly challenging. It wasn't simple. It wasn't certain it was going to work or be successful, but the good news, it was successful. And in 13 years, we were able to figure out how to do it. First, we got organized, then we figured out how to map the genome and get it ready to read out all the letters. And then eventually, we read out all 3 billion letters. And so I just covered a lot of ground very quickly, but I could unpack any of it during the question and answer. But if we look back on the fact that genomics is now a little over 3 decades old, I'm going to now tell you, as this lecture goes along, about the major highlights of this field of genomics in its first 3+ decades. And I just told you about the first one. And that first one was, we read out the human blueprint. We determined the order of those 3 billion letters of G's, A's, T's, and C's for the very first time ever in the Human Genome Project. And that will forever be the #1 success of genomics, because it was such a remarkable thing for humanity to be able to do, read out its own blueprint. Well, let me tell you then, what then transpired, in fact, was a significant amount of interest to then use the fundamental information about our genome to be able to then think about how it can be applied to better understand humans and human biology, but also to eventually use information about genomes to improve the practice of medicine. And of course, this leads to the idea of every one of us has a very similar genome. Every one of our genomes is about 99.9% identical to any other person's genome. So we only differ about 1 out of 1,000 letters. But within those differences between us, in many cases, influences which diseases we may be at risk for, or which things we might be protected for, or in the current environment, just think about it. Why is it some people who get COVID-19 are horribly ill and pass away in many cases, but other people who get COVID-19, they hardly even know they're sick? Some of that is because of genetic differences between us that we're trying to figure out. There was tremendous interest to eventually be able to use genomic information about individual patients to be able to tailor their care better than we do it now. Because I can tell you as a physician, most of the way we practice medicine now is based on how we treat the average patient, not how we treat an individual patient, because we haven't had the tools until very recently to look at an individual person's unique blueprint and tailor their medical care. So when the Genome Project ended now 17 years ago, the idea was, yeah, let's figure out this genomic medicine idea. But believe me, it was very blurry. We didn't really understand what that meant. We just had a general idea. But it was very motivating that especially was what we wanted to do at the National Institutes of Health, where I work, because everything we do is about trying to improve human health. And so by genomic medicine, it is this sort of emerging medical discipline that involves using genomic information about a patient to tailor their medical care, to be able to treat them not on average, but to treat them uniquely based on the particular DNA differences that they have relative to other people. Now that you could define the field, but doesn't mean you know how to do it. And what I could tell you is that became the next phase of genomics journey. First phase was getting the Genome Project done. The last 17 years has been about this journey getting us closer to be able to see genomic medicine be practiced. The starting line for this next journey was the Human Genome Project's completion. That was the end of the project, but the beginning of this next journey. And ultimately, we want to use genomic information to better be able to practice medicine. Once again, like the Genome Project, this was not going to be done overnight. It was not going to be done in one fell swoop with one simple step. It was going to be a journey of many steps, and we wouldn't even know what all the steps were. And it was not going to involve one person or one clinician or one country or one discipline. Rather, and it wasn't going to be done fast. It wasn't going to be a sprint. It was going to be a marathon with, once again, many, many people, lots of different backgrounds, lots of different areas of expertise coming together to try to figure out how do we go from the most basic information about our blueprint to actually change the practice of medicine. So that's the first third of my talk. So let me pause there and just see if there's any questions, because now once we're done with questions, I'm going to now take you what's happened and give you a review what's happened over the last 17 years as we've inched our way closer to genomic medicine. So Marianne, and I think who was the other person, was going to moderate any sort of questions you may have at this stage. So maybe I'll take 3 or 4 minutes to answer some questions.
I have just one question.
Sure.
On how you map, well, I don't want to know like every detail on how you map the human genome, but how do you do it with so many, even though there's like few variations, how do you account for the variations anyway?
So it's a great question. And maybe I could try to even state the question a different way. We're often asked, well, whose genome did you sequence? And because if you're going to pick a given genome, you know, how do you know that's going to be representative of everybody else? And the wording we use, so I'll give you a little more detail. The wording, we refer to it as a reference sequence. In fact, what the Genome Project produced was a reference sequence, a representation of the human genome sequence. But it's 90, keep it in mind, you and I are 99.9% identical. Across our 3 billion letters, we're 90. So even if that reference is different 1 out of 1,000, it's still a pretty darn good reference to sort of organize and to sort of start to build information about. Once we had a reference, we don't claim it represents a person. It just reflects an organizing framework of 3 billion letters from which we then can start to interpret, and we can then compare people relative to one another.
That makes sense. Right. Thank you.
Sure.
Thanks.
Good question.
I got 2 of the same question from
Sure.
2 people in the audience, and they both want to know about the contributions of Rosalind Franklin to the discovery of the DNA helix. So you brought up Watson and Crick.
Yeah. So I brought up Watson and Crick because their names were the ones that were on the paper, but the question is absolutely correct. And in fact, there's very good books written about Rosalind's data was the data that was absolutely used to figure out the double helical structure. It's probably not the best example of good scientific etiquette because she was not given adequate credit. And sadly, she passed away before the Nobel Prizes were given out, and the Nobel Prize rules don't allow a Nobel Prize to be given to somebody who is no longer alive. And so the history will show that Rosalind was not well treated and was not given enough credit. So it's an absolutely fair question. And you know, and similarly, you're right, I showed the picture of Watson and Crick. The other person who was involved in this was Maurice Wilkins, and he was the one that won the Nobel Prize along with Francis Crick and Jim Watson. But Rosalind deserves absolute credit for generating the data and really having the insights to do the experiments that elucidated this.
Thanks. And I have one more double question
Okay.
that two people asked,
Sure.
which is asking about the greatest difficulties that you encountered in mapping the human genome, like how do you start when you're going to map a human genome?
Yeah, no, it's a great question. And let me tell you one other, I could answer it many different ways, but maybe I'll just answer it at the highest level, is that when the Genome Project started, we did not know how to map or sequence the human genome. We had a goal. We had cursory methods, but we had no, I mean, it's sort of we often make the analogy to the moonshot when they said, we're going to put a man on the moon years ago. And they said, you know, we sort of have the beginnings of computers and we think we know something about rockets, but none of the details were known. And it was the audacious challenge of saying, we're going to do something that got smart people and motivated people to figure it out. I cannot tell you when the Genome Project began, we truly didn't know how we were going to map and sequence the human genome. We just sort of took one step at a time, and we just constantly brought people together and strategized, and were willing to throw out an idea we came up with last month if a better idea came along. And so there were just many micro stories of evolutionary improvements that got us to the point where we could actually sequence 3 billion letters. Believe me, when the Genome Project started, there was just no way we had that kind of capacity it was way too expensive to sequence DNA at the time. But fortunately, better approaches came along because people were very motivated. Okay, those were good questions. Maybe I'll keep going and we'll come back to another third. I'll stop again for more questions. So that was the first highlight. And in fact, this whole thing about the cost of DNA sequencing is going to be very relevant because that was the most, you know, one of the most pivotal changes that have taken place since the end of the Human Genome Project. In fact, the second highlight I want to show on this slide really has been overwhelmingly influential, not only on genomics, but on almost all of biology. Because what it really relates to was what I was just alluding to, that the Genome Project was successful. I'm going to skip that. The Genome Project was successful in sequencing the human genome for the very first time, but it didn't come at a cheap price tag. It cost about $1 billion to generate that first human genome sequence, $1 billion. Now, it was worth it, but keep in mind what I was saying. The ultimate goal is to eventually be able to sequence a genome of a patient sitting in front of you and use that information to tailor their medical care. And clearly $1 billion is too expensive. And so when the Genome Project ended, the field really got very motivated at a new goal. And the new goal was to try to remove as many zeros off of that figure. And in fact, we finally settled on the goal would be $1,000. Could we have a $1,000 genome? Could we develop technologies that would allow us to sequence a human genome for what would be a reasonable clinical test? Like, because a CAT scan or an MRI is about $1,000. That seemed totally reasonable. But this was not going to be easy because the way the human genome was sequenced the first time involved big factories like this, these big machines that are the size of refrigerators, each of them costing almost $1 million. There were several dozen of these kinds of laboratories around the world, each of them having dozens, if not hundreds of these machines. This is why it cost $1 billion. What was needed was something very new and very cutting edge. And not only did we need something new and very cutting edge, we also had to become much faster. Because that first human genome sequence, I told you it cost $1 billion, but it also took us 6 to 8 years of actively sequencing human DNA. The Genome Project lasted 13 years, but 8 of those years we were sequencing human DNA to get out that final 3 billion letters. That's not a clinical test. You can't wait around for 6 to 8 years. The good news, and here I'm going to rush through years of progress over the last 17 years, is that we now have methods in hand that allow us to sequence human genomes actually for less than $1,000. And in fact, we can do it very quickly. We can get it done in about a day or two. In fact, increasingly, it's getting down to a day. And so what has happened is a technological revolution over the last 17 years that has reduced the cost of sequencing a human genome by 1 million-fold. Just think about that, 1 million-fold reduction in the cost of sequencing the human genome. That has been overwhelmingly impressive. And the good news is that better technologies are coming along all the time. The latest rage is something called nanopore sequencing. I'm not going to talk about it. You could read about it, but it basically is a very fancy way of pulling DNA strands through a very small hole in a membrane that allows you to directly read out the letters of DNA. And in doing so, you can do it with very small instruments. So shown here, I don't know if this is going to work with my virtual background. I actually have one here. Yeah, I think you can see it. This is actually the instrument that is made that is for nanopore sequencing. And look how small it is. And look, this is a USB port. This just plugs into the USB port of a laptop computer. You don't need a laboratory. Everything you need for sequencing is I'm holding in my hand. And look at this gentleman. This was conducted in Africa several years ago during one of the Ebola outbreaks to sequence the nucleic acid in the Ebola virus. And so this is really miniaturizing and making very efficient DNA sequencing where you don't even need much infrastructure. You just need a laptop computer. That same device was also used in Antarctica for sequencing DNA in Antarctica. And that same device was actually used up on the International Space Station, where DNA was actually sequenced in space by this scientist. So the point is that, oh, and by the way, the same company is allegedly coming out with a device that will attach to your smartphone, your iPhone or your Android or whatever, to be able to sequence DNA. The technologies for sequencing DNA have just gotten better and better and better, and they continue to get better. But more importantly, they've gotten cheaper. And so as a result of that, the fact that we can now sequence a human genome, not for $1 billion, but for less than $1,000 has been completely game changing. Because what it has done is it allowed us to now go in 17 years from having one human genome sequence to now having hundreds of thousands of human genome sequences. Now, why is that important? Well, the reason we want to have, and it's actually probably closer to 1 million at this point, human genome sequences, the question I was asked during the question and answer period, is that any one of us a little different, and as I told you, we differ by about 1 out of 1,000 bases. But if I compare my genome to any of yours, if you do the arithmetic, that's about 3 to 5 million spelling differences between your genome and my genome. But what's also very interesting is that most of those differences between us, it's not like they're particularly rare. They're actually, most of those, if I find a difference between my genome and yours, there's probably somebody else on this Zoom call that has that same difference. Because most differences occur in several percent of people in the world's populations. By sequencing now hundreds of thousands of people and sharing that data, we now have amassed all sorts of databases that tell us about the most common DNA spelling differences that exist, not just here in the United States, but actually around the world. So we're developing inventories of the most common spelling differences and are now using that information to be able to go and figure out how it influences how the genome works. And we've come a long way in 17 years figuring out how the human genome works. And now we're really turning our attention to when there is a spelling difference in a gene or some other part of the genome that influences how that gene works. We can figure out whether that spelling difference is influencing the function, if it influences it in a good way, in a bad way, does it maybe cause a disease, does it maybe protect from a disease, and so forth. So the whole idea of figuring out when our patients have different spelling in their DNA, is it important? Is it biologically important? And then is it clinically important? And this is a huge important area. We're far from solving it, but you can sort of see that until we can empower scientists with knowledge about how people differ in their genomes, we will only then be able to figure out which of those differences is functionally important and which of those differences is then medically important. With better and better ways of sequencing, better and better understanding of how the human genome works, we've also then been able to go and now figure out increasingly which of those spelling differences play a role in human disease. For some diseases like cancer, which is basically a disease where there's lots of DNA changes, the last decade has brought incredible advances in understanding the genomic changes, the DNA changes that underlie cancer. Similarly, for rare diseases, which you may hear a lot about because the news covers this a lot, rare diseases typically involve one of our 20,000 genes having a mutation in it that then confers the disease, like cystic fibrosis or Huntington's disease or sickle cell disease. These are all diseases where one gene has a spelling difference and it breaks the gene and you get the disease. We now know of about 5,000 rare diseases that we know exactly what the DNA difference is, or the DNA differences are, because it often involves multiple differences across different patients. But years ago, we only knew it for several dozen. Now it's in the thousands. And increasingly, those are rare diseases. For more common diseases, hypertension, diabetes, cardiovascular disease, mental illness, we're learning which are the DNA spelling differences that are influencing those diseases. The more common diseases that all of us are more familiar with, because we all have family members with those diseases are a little more complicated because they typically involve not just DNA changes, they typically also involve environmental influences and lifestyle and social influences and so forth. But bottom line is we have gone from having cursory early information about the DNA differences that play a role in human disease, let's say 17 years ago, and now we have so much more insights. Although believe me, there's plenty more to be learned. We've only scratched the surface in many ways, but it has been quite an impressive accomplishment since the end of the Human Genome Project. Now, here's the other thing. If you've noticed, these 5 highlights on this slide take us from the most basic information about the genome to understanding how differences in our genome play a role in disease. But notice I don't have a highlight about medicine at all. And actually, about 10 years ago, when I first became director of the Institute, this is pretty much where I would have to stop my talk because there really wasn't much more to say. But the good news is in the last 10 years, I could put a new highlight on because we're beginning to see medicine change with the use of genomics. And it's just some of the earliest examples. And I'm certain 10 years from now, there'll be even more and better examples. But I just thought I would just really stress the point that we have now moved from a very blurry idea of what genomic medicine is going to be all about to a much better appreciation. It's becoming much more into focus. And I'm frequently asked, well, what do I think are some of the earliest and hottest areas in genomic medicine? The earliest examples where genomic information about an individual patient is being used to actually change how we practice medicine. I often give these 4 examples, and I'm just going to give them briefly. I'm happy to answer questions about them. One is cancer. I just mentioned it earlier. The fact is, increasingly, if someone is diagnosed with cancer, they're going to get genomic information about that person's tumor and have that unique genomic information about that tumor and that patient influence decisions about how to manage that patient, how to treat that patient. Increasingly, we are learning that the reason we respond differently to medication is because there are spelling differences in our DNA that influence how we metabolize drugs. There's this big word called pharmacogenomics. It's just pharmacology and genomics. Every one of us knows of examples either for ourselves or for family members when you were given a medication and it either didn't work or it made you sicker. And the reason is because everybody responds to medication a little bit differently. And we are increasingly learning what the spelling differences are that make you either a good responder or a poor responder. And so, as a result, we're getting to the point where for some medications, we do a genomic test to figure out if this medication is the right one for you or not. And that's called pharmacogenomics. You're going to hear a lot more about that in the next 10 years. Increasingly, when a patient has a rare disease and doctors don't know what is wrong with them, nowadays, unlike 5 or 10 years ago, nowadays they sequence their genome. Costs less than $1,000, and in over half the time you can figure out what is wrong with a patient. And so in many cases, these are patients who have not been diagnosed. They can't figure out what's wrong with the patient. You sequence their genome, about half the time you can figure it out. Many, many stories have come on about this, and increasingly all around the world, this is being done. And lastly, this is also changing prenatal genomic testing, because lots of couples decide to want to have some genetic information about their unborn child. Not all couples, but many couples decide that. But increasingly now, through a method that allows a simple blood draw from a pregnant mom, you can detect using these new methods of sequencing the very small amounts of fetal DNA that naturally float around in mom's bloodstream. And you can get tremendous amounts of insights about possible genetic defects in an unborn child to give the couple information that may be helpful to them. But you're doing this in a very non-invasive way rather than having to do something called amniocentesis, where they have to use a needle and get into the little bit of amniotic fluid, which is an invasive and unpleasant procedure. Now they just could use very fancy methods that just can use with a simple blood draw of the pregnant mom. So and in fact, that's used millions of times around the world now, this prenatal genomic testing. So here are just 4 examples. Other ones are coming on the scene, but genomic medicine is truly arriving now, and it's very exciting. So let me pause there again and see if there's questions before I just go through very briefly the last part of my talk.
There was a quick question from Annika, which is that
Sure.
You're the Human Genome Research Institute, but
Sure.
How can animal DNA sequences maybe give us better information?
You broke up right at the word
animal DNA sequences.
How are animal DNA sequences helpful?
Yeah.
So that's a great, actually, that's something I use when
Yes, sorry.
my research program actually got involved in that. So not only have we sequenced the human genome, we've sequenced dozens, actually hundreds of other vertebrates, other mammals, and we've sequenced thousands and thousands of bacterial and viral genomes and all that. There's a lot of uses in that. One of the things that's particularly useful for us is in sequencing vertebrates is that evolution has a way of not wanting to have DNA change very much. When they're throughout evolution, when something works biologically, evolution tends to keep it the same. So one of the things you could do is if you read the blueprints, the DNA sequence of other related animals, like other mammals and other vertebrates, and you line it up next to us, you can see the areas where things have not changed over millions of years of evolution. And if that's where that's the case, it almost always is pointing to parts of our genome that are functionally important. You know, a good part of our 3 billion letters are not important. We actually don't know what they do, but they're not directly coding for proteins. They're not directly influencing genes and things like that. But what's called comparative genomics, sequencing other genomes, comparing them to ours, is like using a highlighter in a textbook to highlight the really important passages. And that's what we've been able to do by studying the genomes of other organisms. Any other questions?
Yes, so Akshar was wondering, what are some of the controversies that surround genomics in today?
So I think there's a number of controversies that we've seen. In fact, there's a whole area from the very beginning of genomics that we refer to as ELSI, E-L-S-I. It's ethical, legal, and social implications of genomics. And it's sort of a unique feature of genomics of trying to get ahead of these ethical dilemmas. I would say the big issues around genomics right now, there's a helicopter going by, so hopefully that you can still hear me. I'm not that far from Washington, D.C., where there's been military helicopters sort of monitoring what's going on there. Elsie reached some of the areas of bioethics attention in recent days. One has to do with privacy. You know, we want to use genomic information as part of medical care, but we want to make sure people's privacy is maintained about we want to make sure they're not discriminated against based on the particular genomic changes they have in their DNA that might influence their health and well-being. I mean, there's a lot of issues about when people are participating in research and are sharing their genomic information, who can use it, who can analyze, should people be able to profit from it? You also then get into some issues that I didn't really talk at all about genome editing. I talked about genome reading, sequencing, genome editing, and CRISPR, probably many of you have heard about. Lots of ethical issues about modifying our DNA, editing our DNA using methods like CRISPR. So yes, there's a whole host of them. There's nothing more intimate to us for each of us than our blueprint. So you can imagine that there's a lot of ethical issues surrounding with studying people's blueprints.
All right, that actually hit a lot of questions because people were really wondering CRISPR. How is it, Miguel is wondering, how is it possible to separate fetal DNA?
So it's a great question. It turns out that this gets complicated quick. Let me try to give a simple answer. The unborn child from the placenta naturally sheds a little bit of fetal DNA that floats around in mom's bloodstream. When you just take, but there's also a little bit of, but that's of course cell-free DNA. If you draw mom's blood and you get rid of the cells, you just take the liquid part, just the non-cell part, there's some mom DNA in it, and there's also some of the baby DNA. But there are going to be differences between those. And so if you just sequence everything that's there, feed it into a computer, you can actually figure out what's what. And you're basically, there's various fancy things that you can do computationally to sort of separate, sort of figure out which is which. And in particular, it's for the genomic testing, what you're not necessarily trying to do complete perfect sequencing of the baby. You're mostly just looking for abnormalities like abnormal numbers of chromosomes, like the famous trisomy 21, 3 copies of chromosome 21, also known as Down syndrome. So much of what you're doing is making sure there's not an unrepresentative number of sequences coming from a particular chromosome. So as a screening method, this turns out to be perfectly good. So let me go on. Let me just finish the last handful of slides, and then I'll happily answer questions for as long as you guys have time. at least up until the hour, because then I have to scoot. So I just wanted to now take you into the future. I've told you about the story of genomics for the last 3 decades, but I also want to readily admit, I sort of was hinting at it before, that my focus has been around DNA and around the genome. And if we think about human diseases, I make no claim that everything about disease is in our DNA. Only some part of disease causation is in our DNA. But we fully recognize that both genomics and other factors play a role in most human diseases. I have just been really, really lucky. I study DNA, and I have had these wonderful new technologies allow me to study the left half of this pie chart for the last 20, 25 years. But increasingly, we are getting better technologies that are enabling us to study the right side of this pie chart, to measure our environment, to measure our lifestyle, to measure how much we exercise and so forth, what we're being exposed to. And increasingly, some of these things can actually be used and with fancy methods for actually monitoring our physiology. There's all sorts of very fancy things that interact with your iPhone to collect information about things in your blood or how your heart is working, or various chemicals or things that you're making in your body, measuring their levels. And so this has led to this notion of not just genomic medicine, but precision medicine. And genomics was just focused on genomic medicine is just variability in our genomes among people, but precision medicine is just taking a step back and not just focusing only on genomics, but just doing a more precise accounting of individual variability. So in addition to genomic variability, all of us is a little bit different in other ways as well. Our physiology is all a little bit different, and we have better and better ways of measuring our physiology through all sorts of new technologies, including iWatches and iPhones and so forth. Increasingly, we can measure differences in our lifestyle, how much we sleep, how much we exercise. Increasingly, we have technologies for measuring what we're being exposed to in our environment, and increasingly, we can measure what we're eating or what destructive things we're doing to our body. So this has created an opportunity coupling the technologies of genomics with these technologies for measuring these other parts of our existence that play into our health and our diseases. And so the notion is to do research on this now. Just like we've done research in genomic medicine, there's incredible interest in doing doing research in precision medicine, which marries data from genomics with data from all these new technologies. Oh, and by the way, compared to 17 years ago, all of you folks grew up in an environment where all your medical information was being captured through electronic health records. That has not always been the case. 20 years ago, there were hardly any places that had electronic health records. But now, all this data about people are being collected as part of their electronic health records. And the idea is, can we put together genomic data, technologies that allow us to measure these other things of our life coupled with medical data and study this in a very big data way? That is going to lead to a new era of precision medicine. But we got to figure out how to do this. And to do this, we need a big study like the Human Genome Project. And so there's big studies going on around the world. In the United States, the big study is the All of Us Research Program. Maybe some of you have heard of it. Maybe some of you might participate in it. Or what I'm hoping is that some of you will become scientists and clinicians and help analyze the data that's coming out of this over the next 10 years. By the time you get to graduate school in particular, but even during college, there's going to be all sorts of data here. By the time you're in graduate school or medical school, there's going to be an immense amount of data that are going to require creative geniuses like some of you are already becoming to help us analyze all this. The All of Us program is it's all being run by NIH. It's getting 1 million people in America to sign up and be volunteers to be studied, probably for 10 or 20 years. Participants are going to share genomic data, their lifestyle information. They're going to wear all sorts of devices and carry iPhones, and they're going to have Fitbits, and they're going to measure all sorts of things about themselves. And they're also going to give access to their electronic health records. And then meanwhile, we're going to protect that data, but we're going to allow scientists, clinicians, high school students, citizen scientists to sign up to be able to analyze the data. So if some of you go out and get really good skills in data science, help us figure out all these little subtle contributions to health and disease and all these subtle ways that we can improve the practice of medicine by being more precise, not treating patients on average, getting information about their genomes, their lifestyle, their social environment, their exposures, et cetera, et cetera. And that will be a much better way to practice medicine. If you want to read more about all of us, go for it. There's a nice website where you can do that. And there's videos and a lot of, it's really getting exciting now. Just shy of 500,000 people being enrolled, and a lot of data is now being generated. So we're going to get to a day where the practice of medicine is going to be much more precise. We're going to focus not only on G's, A's, T's, and C's of our genome, but other things that are part of our everyday life. So that's the story, the journey. I've taken you from the very basics of genomics to the present time in genomic medicine and given you a glimpse of the future. For those of you who are going to medical school, I guarantee you, you will be practicing genomic medicine and precision medicine by the time you graduate medical school. It will be an integral part of the practice of medicine, I'm completely convinced. Let me just leave you with one additional notion. I have the luxury of representing a field that has a great story to be told. It's very exciting. As you can tell, I'm extremely enthusiastic about it. And I make very pretty slides that make it seem so easy. None of this was easy. None of this was easy. You know, it doesn't go in a straight line. Research is I like the question about the Human Genome Project. You know, did you really, how was it, what were the challenges? This is what the Human Genome Project was all about. This is what the past 17 years was about. This was the journey to figure out how to reduce the cost of sequencing by 1,000,000-fold. This will be the journey to precision medicine. It's always filled with twists and turns. It's the exciting part about research, is that we don't really know necessarily what we're doing. We're figuring it out. It's why at my office at NIH, literally, I have a quote from this guy. Hopefully all you know who this guy is. But I have a quote from him. It literally is above my door in my office. And it's the Einstein quote: if we knew what we were doing, it wouldn't be called research. That's what makes research so exhilarating. I've been a researcher my whole life. And it's because it's not linear, it's not known, it's always a challenge that makes it very, very exciting. And I hope I've shared that excitement with you today. If you liked what you heard and you wanted to stay connected, you want to hear stories of genomics and what's going on, and if you want an extra email every month, my staff and I put together a newsletter that we email out. You're welcome to sign up for it. I won't be offended if you don't, but you're welcome. If you want to get an extra email, we always put little stories together, update you what's happening in genomics. And for those, it took me until I turned 60 years old this past December, but one of the reasons to deal with, I want to do something different at age 60, and so my communications group convinced me to sign up for Twitter. So I now tweet every week, multiple times. If you want to follow me, you can follow me at my Twitter handle, and we try to make it interesting and fun for everybody who's interested in genomics. So that's what I wanted to tell you. And now we can just end the session with any questions. At least I have another 10 minutes if you need it, and I'm going to stop sharing so you can see me. So, and I'm happy to take questions through the chat box, or if people want to ask me directly, I'm fine with that as well.
I put this in the chat earlier, but I asked, is it possible to treat diseases like trisomy 18 and trisomy 21 after detecting them in fetal DNA by altering the DNA sequence before the child's born, or would that be too invasive?
So it's really hard. It's a great question. So the idea is if you're born with an extra chromosome and every cell in your body have extra chromosomes, you know, could you imagine treating it? So I think the issue is, it's hard for me to imagine how you could genetically alter that person, because I don't know how you take away a chromosome. But I will tell you that there are major studies going on, including by one of my colleagues who runs the Child Health Institute, one of the other institutes at NIH, the Child Health Institute. And one of her areas of expertise is trisomy 21. And working with a mouse model of trisomy 21, they're trying to figure out possible treatments to deal with some of the features of that disorder, to try to sort of minimize the bad effects of having 3 copies of chromosome 21. But from a genome editing point of view, that's a tough one, because there's just every cell in the body that has too many chromosomes, and so it would be hard to chase that down.
You really didn't go into this, but I just wanted to know if you could talk about RNAi and like that, your insight on that, and maybe possible uses in the future.
Sure, it's a great question. So not everyone will even know what RNAi is, but bottom line is, I think Anybody who's taken a biology class knows sort of the central dogma, how DNA makes RNA and then RNA makes protein. So there is this whole RNA world, which I didn't really talk about. It's this intermediate nucleic acid that serves a very important function. And there's been discoveries made years ago that basically you could synthesize what are called, well, I mean, actually it happens biologically as well, but in the laboratory you can synthesize molecules that are inhibitory to RNAs in a fashion that therefore will influence the functioning of an RNA molecule. So it basically is a way to jump in at the intermediate and try to dial things up, or more typically dial things down. And that experimentally is actually very valuable for figuring out what genes do and various aspects of genome function. It's also being used therapeutically, because the idea is there are ways of potentially synthesizing an RNAi molecule and introducing it into cells in a patient in a fashion that would sort of mitigate a problem that's caused by some naturally occurring RNA molecule. So I would say is for both an experimental tool in the laboratory and also as a possible, actually a proven therapeutic tool in some cases, it is a very useful thing to have in the toolbox for both doing science of understanding how the genome works and also developing sort of genomic-based therapeutics.
Okay, thank you so much.
I have a
So
question
Marcel was
from the
wondering-
chat. Oh, go ahead, Leo.
Go ahead, Marianne.
Okay,
Yes.
a question from a while ago about the process of going from knowing information about someone's genome to actually making a improvement in treatment or changing medicine.
Sure, of course, it completely depends on the context. So every one of these situations, it's context specific. Let's take an example. Let's take an example of cancer, because it's one of the most common ones. We now have learned that if a patient has breast cancer, for example, that's a very nondescript diagnosis, because we're now increasingly, you could have two people with breast cancer. When you actually look at the genomic changes in each of their tumors, it could say very different things about what's the best treatment option for them. So increasingly for cancer now, we are getting genomic information about the tumors and figuring out based on that signature of changes, whether it's better to go with drug A or drug B. The same is true for lots of other medications outside of cancer. Are you better to get drug A or drug B based on DNA spellings that you happen to have in your natural genome? And another example would be for rare diseases. Patient has some unknown disease, you sequence their genome, you figure it out. In some cases, and when it happens, it's miraculous. You realize you have a broken gene and there's already a drug available to try to fix that problem. And I can cite dozens of examples where patients literally thought they were they would have probably passed away. And you could figure out just by genomics what was wrong with them and figure out a treatment. It doesn't always have a treatment, but it gives a clue about possible treatment options.
So I'm just a mute right now because I want to check with the two things. How's your daily job look like? And what did you do during the high school to get into this field? So before I got the additional question.
I think you're muted.
Scientist. So, that's the first thing is I come from a science family, but I didn't decide to go into science at all until really late in college. And in fact, in high school, I really didn't excel at science in a very serious way at all. What I would tell you is I would say the two biggest influences that prepared me in high school for what I did later was well, we didn't even have AP classes back then, and having now had two kids graduate high school, I think there's way too much focus on AP classes and way too much focus on being an adult when you're a kid. In high school, I found the two most valuable things I did was I had really excellent teachers, a couple of them in English that really helped my writing skills. And then the other thing is I was very active in speech and debate. Maybe you could tell I enjoyed talking. But I think it was the way it organized my thinking that really later helped me professionally. So far more than how many semesters of any science class I ever took in high school or college, far more than any of that, these basic skills in written and spoken English was, I think, the key thing for my professional success. And I learned all the science later. You asked about what's my life on a daily basis, what's my daily routine. I'm not sure there is a daily, it's a pretty crazy existence. And then this COVID situation has made it, many times worse, as is true for everybody in the world. So it's hardly unique to my situation. What I would say is the institute that I run has 650 people in it. So I'm responsible, first of all, for just making sure our organization runs well. That includes people that are active scientists right here on the NIH campus that are doing genomics research. And I have people that help me run everything going on there. But about 75% of the dollars that we get, by the way, I'm responsible, just so you know, all of you and your parents all pay taxes. And your taxes are what fund all the research that we're doing, because we're part of the federal government. And I get about $600 million a year for genomics research. So that's my responsibility, is to make sure that we take this precious $600 million that we're getting from all of you Americans through taxes and are making the best decisions about doing the best science. 75% of those dollars are going out to universities and institutes and centers, actually around the world, but in particular in the US, in the form of grants. And so we're constantly figuring out how to give away this money. There's a whole process for doing this, but it involves a lot of rigorous monitoring of ideas and proposals to make decisions about what money. What that amounts to are lots of meetings, lots of email. And it usually was in-person meetings and conference calls, and now everything seems to be Zoom for now. But a lot of email. So, I deal with hundreds of emails a day and, meetings. I'm pretty much in meetings about, whether it's in person or conference call or Zoom nowadays, 6-7 hours a day. But I love it.
Last question.
Oh, it's a great question. Could you explain some ways genomics is playing a role in COVID research? It's a terrific question. I was hoping somebody would ask that. When the COVID pandemic broke out, at first you could say, oh, well, you're all about human DNA. You're all about human genomics. This is an infection. It's actually an RNA virus. It's not even a DNA virus. It's, it's infectious disease. It's a whole other institute. All of you have seen on the news my good friend, Tony Fauci, who I deal with all the time and who's as wonderful in person as he seems to be on television. He's the real deal. And he's truly one of my mentors. I adore him. I thought this was all his problem, right? Well, no, not at all. Human genomics is absolutely playing a role. I can quickly run through. How is it that human genomics is playing a role in trying to help the COVID-19 pandemic? Number one, all of these testing methods to try to scale up, make better. Some, not all of them, but some of them are analyzing the nucleic acid in the virus, either in the form of RNA or making it then into DNA and then analyzing it. Many of my scientists who I support are actively involved in improving those technologies. And so just the sheer analysis of the viral nucleic acid is being done with the technologies that we have developed. That's one example. A second example is all sorts of synthetic DNA going on and synthetic genomics to try to help do a bunch of things to better make vaccines. And so that'll be very, very important as well. You sort of see it in that arena. The third thing, which is really what we think in many ways is incredibly important and incredibly insightful, going to be really important to understand, is I alluded to it earlier. Why is it that people respond so differently to this infection? Some don't even know they have it, and other people are just, it becomes fatal. So we are standing up very major international study, which you can read about, which is going to involve trying to figure out at a genomic level the people that are good, who respond poorly and who don't seem to have much effect of the virus. There is going to be some very important genomics, human genomics, going to be learned about differences among people with COVID. So those are just three of the examples, and there's others. Maybe one more question if there's time, and then I actually do have to scoot, but I can take one more. Is there any in the chat?
We can take the Marcel's question from before, which is about data privacy, and just asking generally in the Human Genome Project and in other projects, how do you protect individual data privacy?
Yeah, so the first answer is we as a society have to agree what is fair game and what's not fair game. And unfortunately, we have the US Congress behind us because a number of years ago, they passed something called the Genetic Information Non-Discrimination Act, GINA. You could read about it, G-I-N-A, which basically says you cannot be discriminated against based on your genomic information. That's a good first step. It's sort of a civil protection step. Now, it's still not, you know, like any other civil protections, it's going to have to be challenged, and we're going to have to sort of work our way through it. You know, I think us as a society should say your medical information, your private information, such as your genomic information, it should be yours and nobody else's. You should decide who should access it, and people should not use it in nefarious ways. And if they do, they should be pursued through legal action. The truth is, you know, we can do our best to protect information when people participate in studies, and there's efforts to do that, just as there's efforts to protect their medical information. But you know, none of it's perfect, right? We all recognize occasionally our credit card number gets stolen and our social security number gets stolen. And so we also can't overpromise and say, well, we can't 100% guarantee no one will ever get it. We can just say we're going to do our best. So we have to be realistic, but I think more importantly, we as a society, you as a generation, to say, no, we're not going to allow this to inhibit progress in this area. Societally, this is unacceptable. We're not going to allow violations of privacy to take place with people's genomic or medical information. So, okay, Dr. Lee,
Thank you so much.
This was
Thank
fun.
you so much. It was an amazing talk, and we got so much covered. And thank you so much for your wonderful talk.
Absolutely. And let me just end by saying I suspect I'm going to leave Dr. Lee with many, many questions. So I feel badly because I'm sure these students are going to come up with all sorts of questions and they're going to throw your way. Let me make one last point. We do have a lot of educational resources available to us. If you just come to our website, genome.gov, it's really easy to remember. Lots of stuff. And I have a whole education group. And if any of you are interested in learning about more educational opportunities, I could point you to our education group. So lots of stuff is online. So we are very interested in getting all this stuff to all of you and to your teachers and to your friends. So thanks so much. And everybody, again, please stay safe. Listen to your local health professionals. Don't do stupid things. Stay safe for the next, however many months it takes. I know it sucks to be young and be stuck inside. So, all right, take care. Talk to you soon. Bye-bye.